So today we are going to talk about lecture 6, but this is 6A, which is about environmental sampling. And we can see this is a summary and extension of lecture 6B and lecture 6C. And you guys can find lecture 6B and 6C on ELMS. So I recommend you guys to take a look at these 2 lectures. So today we also have 6 topics. For the first one, we are going to talk about the significance of environmental sampling. And then we are going to talk about like planning and sample collection and sample transportation. And after we got the sample, we want to analyze our sample, but we cannot analyze it directly. So we need to process the sample. So we are going to talk about sample processing. And then we are going to talk about sample analysis and data processing. But these 2 are not the focus of this lecture because we have already talked about data processing in statistics. And sample analysis will be talked about in the future unit. So first, let's look at some background. So in the environment, we have pollutants. And mainly, the pollutants are distributed in 4 components. Like we have the pollutants in the water, pollutants in soil, and pollutants in air. We also have the pollutants in the organisms. And actually, I like this animal because it includes both animal and plant. So, and to evaluate the concentration of the organisms, and we usually call this ecological risk assessment. And I will be talk about this in the organic chemistry unit. Okay, I guess. Then let's look at some more backgrounds. As I mentioned before, we have 4 components, and to be more specific, they are they called medium. And since 2003, US EPA and its sister agencies developed a compendium of analytical methods for chemical. 2 people. Wait a minute. For chemical, radiochemical pathogen, and biotoxin analytes of concern. And this is a page of the biotoxin. And you guys can find this information on the EPA website. As we mentioned in the first class, we have a standard method because we want to make sure when different people do the measurement, we can have the same, we follow the same procedure, so the results can be cross-compared. And in support of the efforts of to select appropriate analytical methods, documents to provide information regarding collection of samples of analysis are necessary. So the documents are very important. And in order to avoid analytical errors, it is important to pay particular attention to proper collection and handling of the sample before analysis, which means we need to think about this before we do the experiment. And here shows the whole process, like from sample to data. So basically, we have about 6 steps. The first one is planning. We are thinking about some questions like this. And then we also have the sample collection. While we have a plan, we are going to collect the sample. But for sample collection, we need also think about some of the related questions. And after we got the sample, sometimes we cannot directly analyze the sample on the site. So we need to transport or preserve the sample until it is in the lab and until it is readily available to be analyzed. And then we can analyze the sample. But for most of the situation, the sample need to be processed before analysis. So we need also process the sample. And finally, when we analyze the sample, we can get our data, and then we can do some data analysis based on the results of our sample. And as I mentioned before, we have already talked about the data analysis in the previous lecture. And for the sample analysis, we are going to gradually talk about in the future lectures. So in this lecture, I will be focused on the first 4. And so far, do you guys have any questions? No? Okay. So first is planning. So before we start our project, we need to have a good plan. So here I have a question, like, what is a good plan? Like, how do you guys think? Do you guys have any ideas? Mm-hmm.
A good plan has details.
Details?
Yeah, lots of details.
Yes, so it has details so other people can follow the exact step.
What you're collecting, how much of it, how you're going to contain it.
Yes, so it will have an inventory to list all the stuff that we need. Any other ideas? No. And I think one of another most important thing is that a good plan is economical, like it is relatively cheap. So we can use a certain amount of money to do more work. So about sampling plan, we need to think about 4 components, like experimental design, about like what data will be collected, will we use any pre-existing data, or the site selection. And we also care about the documentations, like the log sheets, notebooks, and laboratory notebooks. And as you mentioned, the custody, and also the standard operating procedure. I will talk about this in the next slide. On the other hand, we need to select the proper method. So generally, we have 2 methods to choose. One is EPA method, and another is from the literature. So I have another question, like which one do you think is better? Or yes?
I would say EPA just because it would be like validated by.
Mm-hmm. So you think EPA is better?
Yeah.
Yes. And how about you?
Yeah, it's gonna be
Same EPA? Yes. How about you guys? No? Okay. So I would say it depends. Like if we want to measure something that is advanced, so we may use the method from the literature because EPA methods may not be that accurate. And actually sometimes EPA method may be a little bit more traditional, so it is not that economic. But on the other hand, the advantage of EPA method is that it is legally defensible. Like for some time, if you have a lawsuit, but your enemy does not understand environmental science, so if you explain the method in the literature, he cannot understand. But if we say we use EPA method when we have the lawsuit, it's better defensible. So it has both advantages and disadvantages. The last one is supplies. So we have sampling supplies and containers, and we need to think about the transportation. Now, I want to talk more about the documentation. And I find this picture is a lab notebook from a Nobel Prize winner. And we can see his or her notebook is very clean, and I like this style. Okay, anyway, so usually we will have the laboratory notebook, and we have the field notebooks, and we have the chain of custody, and we have SOP. Again, I will talk about this later. But we must have the version control, because when we have the procedure and when we are doing the experiment, we may find this SOP can be improved. So sometimes while we do the experiments, we can revise the SOP, but we want to make sure we have the correct version because different people may use the same SOP. Also, the unambiguous sample ID. So we want to make sure the sample ID is readable and identifiable. Like, especially, like according to my experience, when we have some sample ID, like for some sample from many years ago, but for now, I'm going to re-measure that sample. And based on that sample ID, I cannot identify what is that, or where does it come from. So this is also important. And I have another question. Can I use iPad plus Apple Pencil or other electronic devices to take laboratory notes? How do you guys think? No? Generally, no. How do you guys? Okay. Yeah, anyway, I think for now, we cannot, because it is a little bit dangerous. Like if we spread something on our electronic device, it may have some problems. But I would also say maybe in future, we may do this. Like, because if we have electronic device, when we are doing experiments, we can record the whole process, and we can take photos, and that can provide more information than the written documents. And I think maybe in future, we can have some AR or VR or something like hologram, so we can have more information than just the written documentation. No, just in case you spill something. I'm not quite understand.
In response to the question of whether or not you can use your iPad to take lab notes.
Excuse me?
I was saying that, like, the note, just in case you stole something, I think that's in response to your question.
Yeah, that's correct. Thank you. Okay, let's look at next slide. So SOP refers to Standard Operating Procedure, and a Standard Operating Procedure is a set of written instructions that document a routine or repetitive activity followed by an organization. So I think in future, we will not have a written instruction. Maybe we have a video or some other stuffs. But for now, it is a written instruction. And in the homework assignments, I think you guys are asked to develop a SOP. And this is important because it can provide clear information and instructions, and it is consistent so different people can follow the same SOP. And it also provides information about quality assurance and quality control. I just want to make sure if they type or no. Okay. And it usually have several contents like the title, table of contents, procedure, QA and QC, and reference. And here is an example of the SOP of my project. As I mentioned in the first class, I'm doing the soil pollution. And we can see that this is the sampling of DDT and dieldrin in the soil. And this is available on ENCE. So when you guys are developing your own SOP, you guys can take a look at this sample SOP. The next thing to take a sample is to take the representative samples. And the most important thing we need to consider is heterogeneous and homogeneous. And here shows the like the pH value in the soil. We can see that if we are here or we are here, the pH are quite different. So on this side, the pH is heterogeneous. So in this situation, we need to have a good plan to take the representative samples on this heterogeneous site. And I think in lab 4, you guys are going to measure the pH of the soil. Also, we may take some replicates to make sure we got the same results at the same site. And here is another example, like we have the mixed candies. And I think the Halloween is coming soon. And here I have a question. Do you guys think it is heterogeneous sample or homogeneous sample? How do you guys think?
I'd say it depends.
Depends.
Yeah, it's homogeneous when you're looking at like size and shape, and heterogeneous when you're looking at color.
Oh yeah, that's a good point. Yeah, I think that's a good point. And any more ideas? Yeah, the general idea is it depends. And the point I want to show is that, but I think I agree with you, because for me, I only think about the flavor. Yeah, but the shape and the color, they are different parameters. So if, let's say if we want to measure the flavor, so it depends, because if we take the sample one by one, like one piece of candy, another piece of candy, we can see that it is very common to get different colors or different flavors. So in this case, they are heterogeneous. But on the other hand, if my sample equipment is like a spoon, so I can only take the spoons of sample. So I can get a bunch of the candies at the same time. In this case, we can imagine that every of the spoon, their composition are similar. Like they may have some fluctuations, but in general, the spoon of candies, they are similar. So in this case, it is considered as homogeneous. So the point I want to show is that heterogeneous or homogeneous, it depends on the resolution of our sampling technology, or to be more specific, like the spatial scale. So it depends on the spatial scale. Okay, so far, do you guys have any questions? No? Okay. And then let's talk about, like, we need to find the proper method to collect and preserve our sample. And first, we collect the samples in the water. So for liquid samples, like we have different samplers, like the bottles, sieves, brailers, bacon bump sampler, dripper, and the camera sampler. Here I just searched some of the pictures of the sampler described below. And actually, when I saw these prices, and they are very expensive, so I was like this. Yeah, and then for the water samples must be representative of the body of the water from which the samples are collected. So we need to have the representative sample. And usually we fill the sample container within 2 centimeters of the cap and close the container. So basically, we want to minimize the air content in our sampler, because as we can imagine, if the water is in the environment, this is an open system. But if we have a water sample in our sampler, it becomes a closed system. And we can see that from the open system to the closed system, the equilibrium may change. So the composition of the water sample may change. So we, especially with a large amount of air. On the other hand, the pH and the temperature of the water should be measured at site of the collection, because the pH may change and the temperature may change when we have our sample collected from the parent source. And furthermore, the depth of sampling and the appearance of the water sample should also be noted. And again, this parameter may change if we take it out from the parent source. So we need to measure this parameter on site. And the next one is about soil. And this is lab 4. But as I will mention or announce later, so we are going to do lab 4 in the next week. So not in this week. And we are going to do lab 5 this week because of the potential rain. So we have several tools to take soil sample. And this is like the soil angular kits. And sometimes we can have these soil columns. And I like this one. And we can see that from different depths, so this could be 0, and this is later. And we can see that the soil is heterogeneous, like in the vertical direction. So anyway, so we have minimal field preparation, and sometimes we do not preserve the soil. Because for most of the situation, we just go to the site and sample the soil. And we also need to be careful about the homogenization. So basically, if we have like the soil here, for most of the situation, we will mix the soil and consider them as consider it as one sample, and then we measure the average, like the concentration of the pollutants of this whole sample. And soil is usually kept in dark or freeze with free access of air. Again, if we take it from the parent source and if we change its environment, then it will reach new equilibrium. So the composition may change. And if we are interested in the microbial activity, so we are going to take the sample in lab 4 next Wednesday. And this soil sample will be used to measure the organic content in lab 7 and to measure the microbial activity in lab 6. And so in this case, we want to have like this situation. And sometimes prior to homogenization, we need to remove the twigs, roots, leaves, and rocks, and any stuff that is not part of the soil. And you guys are going to do the sampling next Wednesday. And I think after next Wednesday, you guys have more experience on soil sampling. Another part of the environment is sediment. So sediments, we have different samplers. And this is a drench, and this is a gram sampler. And again, this is very expensive. And we can select the sampler depends on the types of the material. And during sampling of the sediments, we usually do not discard any unused sample into the body of the water during the sampling event, because the sediment usually contains more pollutants than the water body. If we just throw it in the water, it may dissolve in the water and release some of the pollutant. And also, we need to decontaminate or re-sleeve the grab sampler as required. Like if we have like several places to do the sampling, when we use this one to do here, if we want to do this place, sometimes we need to clean this one because we want to prevent cross-contamination. Another one is about air. So air sampler, it also has some particular samplers. And here is a bottle vac. So for this bottle vac, we usually just vacuum, like we remove all the air inside the bottle. And then we bring this bottle on the side. And then we just open the channel. And then it will gradually absorb the air from the environment. So we can have the air sample inside this bottle. And sometimes we need an air filter or cartridge because sometimes we want to remove the particulate matters in the air. And sample collection equipment must be assembled outside the contamination zone and prior to sample collection because we want to avoid the sample be contaminated by the chemicals inside the sampling zone. And this is a little bit different from other medium. So for the air sampler, sometimes we need to take notes about its flux. Flux is a function of the flow rate, sampling period, and sampling time. So, like for example, for this one, because we are going to get the total mass of the air, and so the flux is a function of time. So we need to take care of like how much time we expose this sampler to the environment. And as I also mentioned before, my PhD research is about air pollution, and this is one of my projects. So my research is about like the air pollution from the cotton gin. Like we have the cotton here, but if we want to get the cotton fiber, we need to separate like the pure fiber of the cotton and some other thing like the leaves, the roots, or some dust. But during this process, this dust may be released to the environment. So we will have some particulate matter air pollution around the cotton gin. And I just make a graph to be more specific. So this is a cotton gin, and we have the stacks here. And these stacks will release particulate matters in the environment. And here shows a picture of the stacks. We can see that, I'm not sure if it is clear here, but we can see we have some smokes released to the atmosphere from these stacks. And to have the to know the concentration around this whole cotton gin, we have different samplers, and these are the samplers. So basically, we have samplers like we have 3 tiers of sampler. 1, 2, and 3. So we have the samplers or sampling array around this releasing source. And here shows the detail of the sampler. So like this is a 2-meter sampler, it is very short. And we also have some sampling tower, which is a 10-meter sampling tower, like shown here. So for the sampling tower, we have samplers at different height. And in this experiment, we are interested in the total suspended particulate and PM10 and PM2.5. And this is the sampler of the particulate matters. And here shows the internal structure of the, I think this is a PM10 sampler. So basically, we have a pump like absorbing air from this side. And then we have the air will just into this, this sampler. And we have some complicated wind paths. So if we have like very large particle, because of their heavy mass, they will just be thrown on here. But for the smaller particle, they will be carried by the wind, and then they will just go through this wind channel. And then we have a filter here. This filter can collect all the particles that pass through this wind. So in this case, we can design this wind tunnel structure to selectively let the particle size smaller or equal to 10 micrometer. So this is a PM10 collector. And then we have the particles on the top of this filter. And this picture shows a clean filter, and this picture shows the filter that has some particles on it. And because it is near a cotton gin, so most of the particulate matters, they have cotton, something related to cotton. So they are very powdery. So they are not very dark. So anyway, the next thing is about like we others. So another sampling technique is swipe. So swipe are to be collected with care to minimize the damage to the swipe material or introduction of airborne contaminant during the sampling event. So sometimes, for most of situation, we use the swipe to get the microbial samples. And we have large area swipe, and we have the small area swipe. And the total surface area are usually recorded. And except for swipe, sometimes we also need to take the vegetation samples. And for the vegetation sample, we use the scissors and cutters. And sometimes, usually, we do not take the sample plants that are found under large trees or covering of the building unless directed to do so, because this situation does not represent a typical environment of the plant. Like we want to get the representative samples. And we using care to minimize the physical disturbance of the vegetation. Sometimes we do not want to destroy the plant. And when I was preparing for the material, I find a paper from Nature about analysis of the microbial on the leaves. And it is very interesting. Like they found that they have two technologies. The first one is homogenize after subsampling. So basically we have the leaves, and then we can make the subsample of the leaves, and then we homogenize these very small pieces of leaves, and then we do the centrifuge. We have the supernatant or the solution. On the other hand, we can do homogenization before subsampling. So we have the leaves, and then we put the leaves together, and then we have a solution. And after the tissue solution, we mix all the tissue solutions to get this result. So theoretically, they should be equivalent, but they find the different results. I think I didn't read it clear, like comprehensively, but I just find this comparison is interesting. So far, do you guys have any questions? No? Why shouldn't you take vegetation samples that were under latitudes? Oh, as I mentioned before, these samples, they are not quite representative unless we are interested about the plant that's in the specific, like in the particular environment. Can I just put it? Okay. The next topic is about sample transportation and preservation. So we have like the preservation techniques can retard the chemical and biological changes that inevitably continue after the sample is removed from the parent source. So basically, the general idea is that we have the sample in the parent source, like we have one equilibrium. But if we take the sample, the equilibrium will change. So it will gradually change from one equilibrium state to another equilibrium state. And because of this change, the chemical compositions or the microbial content, they will also change. So the sample will not be representative. So generally, we want to prevent or avoid this from happening. And here are some methods of preservation. Like we can retard the biological action and retard the hydrolysis of chemical compounds and complexes. And we can reduce the volatility of the constituents and reduce the absorption effect. So these are our goals, and we have some exact methods. Like we can limit the pH, and we can add some chemicals, and we can also like keep the sample in the very low temperature. So like the microbial activity will stop, or the chemical reaction rates will be significantly decreased. And actually, I think these techniques are pretty like how we preserve food, like preserve the fresh food. After we have the sample, for most of the situation, we cannot analyze the sample directly. So we need to do some treatment to our sample. And here shows the 3 common treatments or the processing methods of the sample. The first one is drying to remove the water, and the next one is filtration. We want to remove the particulates. This is especially for the air or the water samples. And the last one is extraction So we want to transfer analytes into a different solution or phase. For example, like if we are interested in the organic chemicals in the soil or in the water, we can use organic solvent to extract. And for the soil, we are going to do this in lab 6. For the extraction method, we are going to use microwave extraction. But this microwave is not the microwave that we use in the kitchen. So please don't put your food in that microwave. So we are going to use this microwave to do the chemical extraction of the organic pollutant. And then after extraction, one can reduce the volume of the concentrate and analytes, because like usually in the environment, like in the water, we want to measure like the concentration of benzene. We know it from chemistry that the solubility of benzene is very small in the water because this is hydrophobic. So we can just have lots of water sample and we can concentrate it into a very small volume of the sample, maybe by hexene. So we can have relatively high concentration of benzene from this large volume of water, and this concentration can be analyzed by the instrument. So far, do you guys have any questions? No? Okay. The next one is about instrumental calibration. Like finally, we can analyze our sample. And to analyze our sample, we have the calibration. I think you guys did lots of homework and quiz about the calibration curve. We have the concentration and we have the response. And we can construct a linear relation between concentration and response. And here I have a question. And I think this is the same question on the quiz. So you measure a sample with a response of this value. So this value is here. So you got a response here. And the question is, how would you determine the concentration of your sample? So in this case, we can see that this value is outside the calibration curve. And I think most of you think we can just extrapolate this, this line and then plug this as y. And then we can back calculate the x value. So this is not correct. I will talk about this on the next slide. But the general thing we need to do is to dilute, because this is too large. So we want to dilute our sample to make sure we have some signal inside this calibration curve. And then we can do the measurement. If we have some sample that has the response outside the calibration range, then we cannot use that signal to determine the real concentration. And the reason is here. So this plot, this is amounts or moles, or we can understand it as a concentration of our sample. And this is a measured intensity, or we can consider it as the signal strength. And in this case, we can see that the real functional relation is like this curve. So if the concentration is very large, then it will, like the change rate will be slowed down. So in this case, we can consider if we have a calibration range here, and this is almost linear, but if we have the signal that is very large, then it will be something here. But if we extrapolate our linear regression, we will have the concentration here. We can see there will be a very large difference. So once we have the calibration curve, we need to make sure the signal is inside our calibration curve. I also want to use this picture to talk about LOD and LOQ. So basically, when we are using the instrument to do the analysis, we know that the signal will not be stable. Like we will have some background noise, or the white noise. So the signal will not always be stable. It will have something like this. So once we are measuring our sample, like for this example, when we got the sample, we will have very large signal. And in this case, the strength of the signal of our sample is significantly different from the white noise. And in this case, we can know if we have a sample here, and if it is in the calibration range, then we can determine the exact concentration of our sample. But in this case, we can see that we have a signal here. This is very small, and it is not significantly different from the white noise. So in this case, we have no idea about its real concentration. Or we can say we have no idea if this signal is due to the sample or just because of the white noise. So in this case, we don't know any information about the concentration. So in these two cases, we have like two concepts. So the first one is limit of detection. So it represents the lowest amount of analytes in the sample which can be detected but not necessarily quantified as an exact value. Like for this situation, we have the signal that is significantly different from the white noise. But this may be too small that it is not in the calibration range. So we know that it is significantly greater than the white noise, but we cannot say, we do not know its exact value. So this is LOD. We also have the limit of quantification. So it refers to the lowest amount of analytes in a sample which can be quantitatively determined with suitable precision and accuracy. So basically, this refers to we can understand this as the minimum range of the calibration curve. And again, we have within the calibration range. Okay, so for this one, do you guys have any questions? No? Okay. The next one is, once we have the data, we need to data processing. So first, we need to check the data. Especially we want to check if the data is reasonable. Like if we have a pH value which is negative, we know that something must be wrong. And we also need to check QA and QC. I will talk about QA and QC in the next slide. And we may also need to do some unit conversion or transformation of our data. And we make the plots, and we may do some data analysis. And then, based on the statistics, we can get some conclusions. And most of this has been talked in the previous lectures. And so for now, I'm going to talk about QA and QC. So QA refers to the quality assurance. It is generally refers to a broad plan for maintaining quality in all aspects of the program. And this plan should describe how you will undertake your monitoring effort, like the proper documentation of your procedures, training of the volunteers, study design, data management and analysis, and the specific quality control measurements. So basically, QA refers to anything that we can do to make sure our sampling analysis is reliable. And we also have quality control, QC. This consists of steps you will take to determine the validity of specific sampling and analytical procedures. So basically, during QC, we are going to evaluate some of the parameters, like the precision or the accuracy of our data or our method, to make sure they are reliable. And the quality assessment is your assessment of the overall precision and accuracy of the data after you have run the analysis. So during the experiment, we will measure the concentration of our sample But in the meanwhile, we will have like the control, the blank, the spike, and any other stuffs. I will talk about this in the organic chemistry unit. But we have some concepts like the parameter, like the field duplicates and field blanks. They are not, I'm not going to read them one by one, so please take a look at this after class. So we have different parameters, and these are the definition of these parameters. And also this is the meaning of these stuffs. So we have field duplicates, field blanks. We also have method blanks and laboratory control, or we may call spike, and the matrix spike and surrogate spike. So I will talk about in the organic chemistry lecture, but you guys need to like have some general idea about what are they. Also like the initial calibration, verification, and the detection and the quantification limits. So all of this should be reported. And I think that's it. So let's just do a quick review of what we have talked about in this lecture. So first one, pollutants can exist in multiple mediums like soil, water, air, organism, and sediment. Yes, I didn't mention it in the second slide, but yeah, generally we also have the sediment. And before we get started, we need to be well prepared. So the 4 elements then need to be considered, include during the planning. Do you guys have document? Documentation and excuse me? Oh, sorry?
Your design.
Oh yes, design. And you? Selection? Yeah. Method, yes. And the last one? Supplies, yes. Supplies. Okay. And it is important to have what sample? Representative, yes. And the common sample processing techniques include. We have 3. Do you guys remember? Dry, yes. Drying. Yes, filtration and extraction. And what refers to the lowest amount of analytes in a sample that can be detected? The limit of detection, yes. Or L, yes. And the lowest amount of the sample, so this is LOQ. And QA refers to quality assurance, and QC refers to, yes, assurance. Okay, my handwriting is bad. Quality control. Okay, that's good. So this is an announcement that I want to say, because it will potentially rain tomorrow. So we are going to do lab 5 tomorrow, and it will be the virtual lab. So we need to use Zoom, like everyone use Zoom to do this. And lab 4 will be next week. But if it rains in the next week, then we will find another plan. But hopefully it will not rain. And also the assignment 3 and quiz 3 will do tomorrow night. So that's it.